Find the tray and comb in the drawer, and the gel box in the cabinet next to it. You can clean the combs if necessary by spraying them with some millipore water and wiping with a Kim wipe. Align the combs until they are symmetrical. To tape the tray, fold a piece under so you have something to grab later when you're taking it off, and carefully align the bottom of the tape with the bottom of the tray. Next, press the edges down firmly with something like a pen so you're sure that it won't leak when you're casting the gel. Add a second piece of tape around the lower edge and repeat the same process. To ensure that your gel runs correctly, insert the combs exactly parallel to the top of the tray. Also make sure that the combs are not touching the bottom of the tray or your samples will leak out. Find a 125 milliliter Erlenmeyer flask. Next, find a weigh boat and zero it out on the scale. Locate the low EEO agarose. Weigh out 0.5 grams of the agarose. Add the powder to your flask, making sure to get up as much as possible. Find the 50x TAE, and in the drawer below, find a 5 milliliter Cereal pipette. Insert the glass pipette into the plastic handle. Draw up 2 milliliters of the TAE by pressing the top button on the handle. To release it into the flask, press the bottom button. Make sure you dispose of your pipette in the proper receptacle. Get about 100 milliliters or so of millipore water by pulling forward on the trigger. Find a 25 milliliter pipette and use it to add 48 milliliters of the millipore water to your flask. After adding these ingredients, weigh your flask and write the weight on a small piece of tape. Find the orange heat-resistant gloves. Make sure your solution is well mixed, then put it in the microwave and set the timer for 2 minutes. Make sure you're wearing the orange heat glove and swirl the flask in the microwave before taking it out to prevent it from boiling over. Take out the flask every 20 seconds and swirl it to mix the contents and ensure an even formation of the gel. Once your solution is mostly clear, start microwaving it in 10-second increments. You'll notice that there are floating clumps of material. You want to microwave it until it becomes entirely clear and no clumps are left over. Carefully weigh your hot flask. Add millipore water until the weight matches what you wrote on the piece of tape earlier. Find the appropriately labeled aliquot of ethidium bromide in the right-hand fridge. Back out on the bench, add 2.5 microliters of the ethidium bromide solution to the solution in your flask. Put on the heat glove and swirl the flask carefully to mix. Make sure you put the ethidium bromide back in the refrigerator. Get a thermometer and place it into your solution. The solution must cool to 60 to 65 degrees before you pour it into the tray so that you don't warp the tray. Pour the gel solution in carefully and try to avoid creating bubbles. If a bubble does form, move it to the edge of the gel with a pipette tip so that it doesn't get in the way of the samples that you load into the wells. If you ever use a big gel tray, make sure that you level it with a bubble level to ensure even gel formation. Also, try to keep the gel tray on a white bench cover just in case it leaks. Let the gel cool and solidify for at least half an hour. Pour some 1x TAE onto the gel to make it easier to remove the combs. Remove the combs by gently wiggling them and pulling upward. Remove the tape from both sides of the tray and carefully place the tray into the gel box with the wells near the negative electrode. Fill the gel box with 1 times TAE until it just barely covers the top of the gel. Cut a 2-inch piece of parafilm. Find the blue loading die and make 1 microliter spots on the parafilm. Make as many spots as you need for samples that you want to add. Find the tube of millipore water. Add 8 microliters of water to each spot. Pipette up and down gently 3 to 5 times to thoroughly mix the dye and the water. Repeat for each spot. Ask someone about the best size marker to use and carefully load 2 microliters of the latter into the leftmost well that you want to use. Make sure you record in your lab notebook page which well you put which sample into. To load your DNA samples, set an L20 to 10 microliters. Then, using an L10 set to 1 microliter, pipette up some of your DNA. Place it on the spot, then remove the tip, put the tip onto the L20, and then mix the sample up and down 3 to 5 times again. Fully pipette up the sample, and you're ready to load it into the gel. When you're loading your samples into the wells, make sure to squeeze out any air that's in the tip before you put it into the well. Also, be sure not to poke through the bottom of the well when you're loading your sample. When you finish loading your samples, find the lid to the gel box and plug it into the appropriate poles. Then take the cords and insert them into the power supply. Make sure that the red cord is plugged into the appropriate place and that the black cord is plugged in directly beneath the red cord. You cannot have them inserted diagonally. Then turn on the power supply and adjust the voltage to 100 volts. Never touch the buffer while the power is on or you will be shocked. Press the black button and make sure the machine is generating amperage. You can also check whether the machine is working properly by looking for small bubbles. When you finish running the gel, make sure you turn the power off and disconnect the cords before you remove the lid to the gel box. To remove the tray, carefully grab it and hold the bottom of the gel so it doesn't slip out. Tilt it upward to drain off the excess buffer. Open the imager cabinet and carefully carry the tray over. Slip the gel in vertically. Turn on the UV light by pressing the button. On the computer, click the Acquire button. You can then save the image as a .tif and put it on the flash drive. After you've acquired and saved the image, take the gel out and set it back on the tray. If you need to run it for more time, you can put it back in the gel box and run it again. But if you're done, make sure you spray the inside of the imager with distilled water, then dry it thoroughly with paper towels. If you forget to clean the imager, you will ruin the inside. When you're all finished running the gel, take it and slide it onto an old pipette box lid. Take a razor blade and carefully cut the gel into sections small enough to fit into a flask. Make sure to use the flask that you used to make the gel because you want to keep that weight. That way the next time you heat the gel, you'll know how much water to add after it's lost in evaporation. You can also just break the gel with your hands. Add some water to the flask to keep the gel from drying out. Then to seal it in, take some parafilm, stretch it across the opening, and put in a twist to tightly seal the flask. Find the large glass funnel and insert it into the lid of the 1XT AE jar. Take the gel box and carefully pour in the excess buffer. When you're all finished, remember to rinse the tray, combs, and any other things that you used with distilled water to clean them. Set them on the drying rack to dry.